Hello, I'm Alan Rockefeller, and I do a lot of sequencing of mushrooms that I find in California and Mexico. And today I'm going to show you how to edit these trace files and turn them into useful data. And so first we're going to download the traces from GeneWiz or whatever sequencing facility you use. And then we're going to use SnapGene to load in the trace file and see if there's any errors in there. We'll verify that the trace is good using BLAST. And then we will submit that to GenBank so the data is available for everybody else forever. And then we can look into how to generate phylogenetic trees to turn your sequences into useful data. So to start, we got here's the GeneWiz site. And so you can download, here's the different sequencing runs that I have. And if we view the results on a sequencing run, you have, here's the three sequences I have. And you can either get an SEQ file or a trace file. Now the SEQ file is just a text file that has the sequence that's been decoded in it. So what we want to do is get the trace. And so the trace is called a chromatogram. And we will open up the trace with SnapGene Viewer. And so this is a sequence that I made when I was doing a DNA sequencing workshop in Michigan a couple weeks ago. And it was a hygrocebi. And there's a lot, hygrocebi is a genus of mushrooms where there's a lot of new species in it. So there's a lot to discover there. So it's a really good one to sequence. So if we open up the sequence here, and make sure we can see that well. I think I'm gonna let's see. I need to add the window to the stream 1 second. Alright, here it is. So here's the SnapGene Viewer, and SnapGene Viewer is a free software that's really good for editing chromatograms. You know, the regular SnapGene costs a lot of money, but just for looking at chromatograms and fixing them up, then there's no reason to use the full version. So the viewer's fine. So here is this hygrocebi, and if you see at the beginning of every Sanger read, you get a lot of garbage, and that's just when it's just not giving you anything useful. And then you start to get useful data, and after a while these ends go away. And if you look in here, now you have really good data. And good data means nice, reliable peaks that are nice, and one peak in each spot, no double peaks, no garbage in the background. So what we'll do now is we'll just kind of scroll through the read and see how it looks. And this read looks really good so far. So the primers I used in this is ITS1F and ITS4B. And 1F stands for fungal, so it only amplifies fungal DNA. And the 4B stands for basidiomycete. And basidiomycetes are a group of fungi that the mushrooms are in, which means it ignores mold DNA or any kind of other weird DNA and pretty much only amplifies mushroom DNA. So that gives us a read that's almost 800 bases long. And you can see here at the end, it's back to garbage again. So usually I'll cut it right where the ends start. Here I can see this one is a C, so we can change that to a C. But it doesn't really make any sense to try to, you can see the next one's a G because it's black, and the next one is a T because it's red, but it doesn't really make sense to try to get more data out of here. Partially because the reads are not accurate at this end, and also, more importantly, these are the conserved areas where the primers bind, so they're not really useful for the phylogeny or barcoding, so we just want to get rid of this data. So, delete that, these bases at the end, and then we'll go back to the beginning. And so here are a couple ends here, and I can see that this is an A and a T. So I will fix these. I could also just chop it there. That would be fine too. In fact, I think I will trap it because this is another conserved region and there's really no reason to guess and maybe get data that's not solid. So I'll delete off of that end. And now we have all this really good solid data in the middle. So next thing to do is to check this data, and the way I check it is I compare it with all of the closest matches in GenBank. So I'm going to select all and put it on the clipboard, and then we will open up Firefox and see if this works here. I think we want to put this in the background. Let's see, or maybe I can just move Firefox up. Yes, that worked. So now here is Firefox, and what I want to do is go to NCBI Blast, and to do that I will just we can just Google NCBI Blast. So NCBI Blast, Blast is called the basic local alignment search tool. And what this does is it's a way to search all of the sequences that are in GenBank, and it does that using an algorithm called Blast. And so we input a query sequence in here, and it compares this query sequence with every other sequence that's ever been uploaded to GenBank. So here's the query box. A little bit is cut off on the left, I can see, but I think that's fine. It doesn't really matter. And so I just pasted the DNA that came from the chromatogram right in there. So that's about 790 characters or so. And now looking here, these are the defaults on the blast parameters. And you can see right now it wants to search the human genome. And that's not good because our mushroom is definitely not a human. That's not going to work. So what we want to do is search the nucleotide collection. So searching the nucleotide collection, I'll search all of the mushrooms, all the plants, all the animals, every DNA sequence that's ever been uploaded to this nucleotide collection. And this, so I just scroll down and click Blast. So this takes usually about 15 seconds. And so this is comparing my sequence with every other sequence that's ever been uploaded to GenBank. And so people from all over the world upload sequences, and anybody can upload sequences here. So we're searching through a huge number of sequences. And occasionally the server is busy, and it takes longer. But usually doesn't take too long. Usually not more than 30 seconds. Here it's probably taking a little longer than normal because I have kind of a long sequence because the ITS4B primer is a little bit further than the primers people usually use. But here it's finished. And so looking here, we can see our alignment scores in these red bars. And what this shows is that we have a whole lot of returned sequences in our query. You know, their default parameters ask for 100 matching sequences, and that's what we got. But out of that 100, these red bars show how well our sequence overlines with all the other sequences. So usually when people sequence mushrooms, they use a primer called ITS4 as the reverse primer. So ITS1 and ITS4 gives you about 600 bases. So these sequences here are the ones that used ITS4 because it stopped copying at base 600. Now I used ITS4B, so I get a little bit more data in here. And so it stopped copying over here. So you can see that the top couple query matches here, they have their longer sequences, but then they quickly go down to the shorter sequences. But 600 characters is really a lot of data. It's like 600 character states with 4 possibilities. So every mushroom will have a unique barcode in here. And we don't really need all 800 to be confident of what we have. So scrolling down, here we have our matches. And right now it's sorted by total score, but you can click on any of these different columns to sort by whichever column you want to sort by. And the most important ones to look at are query coverage and percentage ident. So query coverage is how much my sequence overlaps with the sequence we're comparing to. So 100% is really good. It overlaps all the way. That means our sequences are either the same size or the sequence I'm comparing to is larger than my sequence. And if you go down to here, you see this one is 81%. And that means they use the ITS4 primer, so they don't have quite as much data, but still that's plenty. And you can see that if we want to try to figure out what my mushroom is, Hygroscopy flavescens is what the blast matches are saying. Now these names you have to take with a grain of salt because anyone can upload sequences to GenBank, and anyone can put any name on any sequence they upload to GenBank. So when I try to upload a sequence to GenBank, I try to put the best name I can on it, but sometimes I learn later that I didn't have, I didn't have the right name on it, so then I'll send an email to GenBank and update the name sometimes if I remember. Though most people just submit sequences and kind of forget they're there, so there's a lot of misnamed sequences in GenBank. And so these actually look like they're named pretty well. So the query coverage, if it's pretty high, like 80% or 100%, then that's good and it means you're going to get a good match. But if the query coverage is really low, like 40% or 20%, here's some that are 67%, that means only, or like here, 53%, that means only 53% of your sequence is overlapping the other sequence that you're comparing against. And when you're only comparing half your sequence, you can start to get errors in here, like maybe some other mushroom has a conserved region, and so you're kind of comparing these conserved regions, but knowing that you're only looking at 53%, the query coverage is really important. And here there's some query coverage of 39%, so when you have it that low, you know that this is not really a good answer. So keeping in mind that the query coverage should be high for a solid answer, I like to sort by the ident field. And ident is counting how many, what percentage of the bases in your sequence match the other bases in the reference sequence. So sorting by ident, the top matches come to the top, and then you just have to know that you got to keep in mind that the query coverage has to be high, because normally it sorts by max score, which takes into account both query coverage and ident. So here's the top match, which only has a query coverage of 81%, and that is overlapping, our ident in that is 99.84, which means there's only one base difference between that and the other. But I think we want one where the query coverage is 100%, and so the next one, the first one where the query coverage is 100% but has really good ident is this one here. And this is uncultured fungus clone, which means that they just sequenced a whole bunch of soil. And their soil had hygroscopy DNA in there. So we'll click on that, and we can see the differences down here. And the way this works is there is a pipe character between the top query, that's my sequence, and then the subject is the sequence we're comparing against. So these characters are called pipes, and there's a pipe character whenever my sequence is the same as the sequence we're comparing against. And when the sequence is not the same, then the pipe character is missing, and it helps you spot the differences. And a good trick, if you want it just to show you all the differences, because sometimes it's hard to spot the differences just by looking at these all with your eye, is you search in the web browser, you hit Control F, and you search for pipe space. And when you search for pipe space, it's looking for places where you have the space character after the pipe, which means that that's the difference. So here it's showing us, here's another difference here. But this one is 758 out of 767, which is actually relatively different. It's probably closely related species. So I think what we'll do to verify our sequence is just look at the top match there. And so the top match, knowing that the query coverage is only 81%, 620 out of 621 bases are exactly the same. So that means there's only one difference, and here it is. I have a C where they have an A. So what I should do is check that spot in my chromatogram and see what exactly was going on over there. See if that's some kind of mistake, some sort of sequencer error, or maybe there's a bunch of copies of the ITS gene in the genome and they're not all aligned. But I want to check that and see what that looks like in the chromatogram. So going back to SnapGene, we just hit Control F to find, and what I did is I copied the sequence right after the part where it was where there was a difference. So I'll hit Control F to find in this, hit paste, and here it is. This is what I searched for. So right here, this C is the base that was different, and you can see the C is a really good read. There's no problems here. So what I was looking for is you saw in the alignment here that I got a C where they have an A. So I was thinking that maybe right here there would be a C and an A on top of each other, and that would indicate there was some kind of problem, some kind of error. But that's not what I'm seeing. What I'm seeing is a really good solid C there with no noise, nothing funny about the shape at all. And that means that that's a good read. And so my sequence really does differ from the sequence, most closely related sequence in GenBank by that one base. And a lot of people always ask, like, how many bases does it have to differ in order to call it a different species? And that's a very subjective question because species are subjective, and you can't really. It's like a human species, concept that goes on top of nature. But if there's a lot of differences and the sequences are clean, then you know for sure that it's a different species. If there's just a couple differences, 1, 2, 5, then it's a judgment call, and I look at other characteristics, not just the sequence. So in this case, knowing that there's the 620 of these bases match, we know that we got a good quality read. So the genes that we got, this is the ITS1 right in here. And then in the middle, there's a 5.8S gene. And then at the end, there's ITS2. So the 5.8S gene in the middle here is a very conserved gene. So I know that if I see differences in 5.8S, that's because there's a problem with the sequence. Because it's pretty conserved, there shouldn't be any differences in the 5.8S. But since there's no differences here, I know we got good reads and we're actually getting good data. So the next question is, what is it that I actually matched? So we matched with KF291-175, and we can open that up and see what that is. And that is a sequence of Hygrostobi flavescens, and it's from Vermont. So my mushroom was collected in Michigan, and now I know that my mushroom from Michigan occurs in Vermont too, the exact same thing. Also, a lot of times you look and see who did this data submission here. For example, this was submitted by Jean Lodge. And Jean Lodge is actually coming over tomorrow. Here, we're going to work on some stuff. But she is the world expert on Hygrostobi. So I definitely trust her name on here. So I know that Hygrostobi flavescens is a good name for this. So now we've checked to make sure our sequence is good quality, and we know what it is. So the next thing we want to do is to submit it to GenBank. And so to do that, we need to have our data in a FASTA file. So I will go back to our chromatogram, select all, and copy that to the clipboard. And now I'll open up a notepad. And I will add the notepad to the sharing so you guys can see my notepad. And the notepad is almost here. There it is. Okay. And now you can see my notepad. So what we're going to do now is we're going to make a FASTA, because GenBank wants to see the sequence in FASTA format. And FASTA is cool because it's very easy and it's human readable. So FASTA always starts out with a right bracket like that, and then we give it a name. And the name actually doesn't really matter very much. But if we're submitting just one sequence to GenBank, the name here doesn't really matter. If we're submitting a whole bunch of sequences at once, which is a good idea if you have a lot of sequences, then the name does matter. But for now, it doesn't matter what we type here, but we do type a name. It has to be something. So hygroscopy flavescens, and then you hit enter, and on the next line you paste the actual sequence. And there's the actual sequence. So now we have a valid FASTA file, and if we wanted to, we could type another caret and put another sequence in here, and we could put hundreds, even thousands of sequences in here, but we're just going to submit this one sequence to GenBank right now. So we're just going to have this simple FASTA with the name and the sequence. And so save that. And I think they're going to want to call that a .fas. So saving it as .text is not going to be good because that'll confuse GenBank. So hygroscopy flavescens, paste, and now we have to save it as, we'll call it flavescens.fas. And it is not a text, it's a FASTA, which is a text format, but they're not going to want to see the .txt on there. So now we have a file called flavescens.fas, and we have to add it to GenBank. So we'll close that, and we will go back here, put Firefox back on top, and here's the GenBank submission portal. So there's a bunch of different ways to submit stuff to GenBank, but the easiest way is through this GenBank submission portal. And so I just logged in here. Anybody can create an account on here. I just signed in with my Google account to make it easy. And so now if I scroll down, I can see the submissions that were here that I've submitted before. So you can see here's the last one I did, a lycoperdon, and there's just all the stuff that I've submitted to GenBank is there. So make a new submission. I'll hit new. And the first thing we do is we tell GenBank what kind of sequence it is. And this is a ITS sequence. ITS is the universal barcode gene for fungi. It's the internal transcribe spacer. So we tell them that it's ITS. And down here, these are different parts of the ITS. We just do this so it detects it. And this title doesn't matter. It's just for me to keep track of my submissions. So I'll say hygroscopy flavescens from Michigan. And we'll continue. And now we can put in some data. This will come up in the record whenever people see your GenBank record. So you can put in your lab. If you don't have a lab, you can put in your house. You can put in really whatever you want here. So defaults are fine there. And now it wants to know a little bit about the sequence. So this was a Sanger read. And this is just a forward read. It's not like I stitched some reads together. So it's an unassembled read. And some people want the sequences to say secret until they've actually published something, but I don't want that. I want my sequence to go public as soon as possible because I'm not worried about anyone scooping me on this. So release immediately following processing. And processing takes a week, so it'll take a week for this to actually get into GenBank and show up in other people's BLAST results. Here's where I give them the FASTA file that I just generated. So I think I called it flavescens.fas. Here it is here. And we'll continue. And now what it's going to do is it's going to run a program called ITSX in the back end. And ITSX processes the ITS sequence, and it looks for certain motifs that it knows should be in there. So it knows for sure that you actually submitted an ITS sequence. And so if you give it some other sequence that's from some other gene, it'll give you an error here, and it'll say, no, I know this isn't an ITS sequence because those conserved regions aren't there. But here it just gave me a warning saying, warning, you're only submitting one sequence at a time. If you're going to submit a whole lot of sequences, you can put a whole bunch in the FASTA and do a whole bunch. But for this demonstration, we're just submitting one sequence, so that's fine. So I'll just hit continue here. And now we have to put in a required field. And these required fields are pretty important, and you get a choice here. But if we go back to our BLAST results over here, and if we go down, you can see the required field there shows up right here in the BLAST matches. So, for example, I think I saw one of my sequences in here. Yep. So my required field I set to the isolate, and for the isolate I put mushroomobserver.org/308214. And that way, that ties the photos that I have of this organism with the DNA sequence. So somebody going through these BLAST matches can say, oh, this guy's got photos. And so if we go to 308214, we can actually see which mushroom this was and where it got collected and all sorts of stuff. So this was Hygrocebi flavescens, and it was from Jackson's demonstration forest up in Northern California. And this was collected at a foray, so we can see, yep, that's the one. So if you have pictures of your organism, it's a really good idea to put in, upload your pictures to a website, a citizen science website like Mushroom Observer or iNaturalist. And then everyone will vote on which kind of mushroom you have, but more importantly, you can tie your GenBank record to these photos. Now this one here I have not uploaded to iNaturalist or Mushroom Observer yet, but I think I definitely should. Should definitely do that because most of these records do not have photos, and it's really valuable to do that. So I'll just make a record for this really fast. And what I'll do is I'll create an observation. I'm just doing this to get the number because I don't have to put in all this data entry right now. So for now I'll just create a blank observation, but that'll give me a number. And the number I got was 389,226. So back over here, our required field will be isolate. And I'll just copy that to the clipboard. And so now we can put in, here's what's required. So definitely the most important thing, or a super important thing, is the organism. And if you don't know what organism you have, that's fine. You can just put in like fungi or some higher level classification. But I know this is Hygrocebi flavescens, so that's what I'm going to do. And isolate, here's where I put in my my mushroom observer number, and this is what actually you see in the BLAST results. And so now I have my unique identifier in there. And now we have the option to add all these other source identifiers. And you definitely want to put some of these in. I would say the most important one is country. Because a DNA sequence is a whole lot more useful if you know where in the world it came from. Because if you add it without that kind of information, then people just don't know, where you got this, and they can't do any kind of analysis as to oh, this is a different thing in one country than a different country. So the country field is USA, but USA is not very specific, so what people usually do is use this country field as more of a general location field. So it's USA, we can put in Michigan, and the city I collected this in was Dalton. And it was collected at a mushroom event at the Circle Pines Center, which is a specific kind of campground place. So we'll put Circle Pines Center. So you can get as specific as you want there in that country field. Isolation source. Host is usually not used, but isolation source can be used as saying what kind of stuff you found it in. So this was just in leaf litter. So this is kind of a free-form field. You can just type stuff in here. Growing leaf litter. And collected by is good field. And I don't have the latitude and longitude right now. I could look up the latitude and longitude of Circle Pine Center, but it's not super important. And the collection date, I don't remember which day it got collected, but that's okay. Um, Actually, I think it was around, yeah, it was like the 29th of September. So it's real picky about formats. So you got to type 29 SEP 2019. If you type the date in any other format, it'll get mad at you. Um, and now a lot of this stuff gets less important down here. Like we can tell it the forward primer we used. My sequencing forward primer was ITS1. I don't really know why you would need to tell them that, but there's a space for it, so sometimes I fill it out, sometimes I don't. Our reverse primer was ITS4-B, and I think that's enough. So I'll hit continue. And now it is, and now here we get to put in a little bit more information about who actually generated the sequence. And a lot of times people submit sequences to go along with the scientific paper. But it's really important to submit all your sequences to GenBank just to make more data available to other people that are working on these things. So I submitted a lot of sequences that do not go with the paper. So we just say unpublished, and then you can just leave this stuff blank and hit continue. And now we just look it over, see if everything looks reasonable, which it does, and click submit. So now this sequence has been submitted, and in 1 week it'll go live, and if anybody else finds the same mushroom, they will be able to see that I found it in Dalton, Michigan. One of the coolest things you can do with this data is to build phylogenetic trees. So I will show you how to do that. So back here, here's our blast. And what we can do over here with this checkbox is start selecting sequences. And I think our top sequence, we definitely want to select that. And these all look like they're the same species because they have the same ident. This one might be a really closely related species called Hygrocebi chlorophanna. So I'll throw that one in there. And basically I'll just go down here and select whatever sequences I want to see in my tree. And here's another one that's claiming to be Hygrocebi flavescens, but this one is only 98% the same, so maybe it's a different species, like a sister species. Here's my sequence from California. You can see my sequence from California is also going under the name flavescens, but it only matches 93%. So 93% is very far, and that means that these are not the same species. And that's why I put the CF in there. The CF means confer to, which means that I'm going to use the name Hygrocebi flavescens because I know that there is no name that matches better, because the one that I found in California is an undescribed species. It has no scientific name yet. But I definitely want it in my tree. So I selected that one. This is probably a different Hygrocebi over here. We'll select this one. Hygrocebi chlorophanna. Hygrocebi flavescens is a name from Michigan. That's where it was discovered. So adding sequences in here from Michigan is really important, because that's if you get the sequence from Michigan, you can be really sure that you're getting the right name, and you actually know what the real flavescens is, which is pretty important. Here's another one, Hygrocebi citrovirens, chlorophanna. So we'll just add whatever seems interesting here. Now, Hygrocebi conica is a black staining Hygrocebi, and that is in a different section of Hygrocebi altogether. So I'll select that, and we'll use that as our outgroup. And an outgroup is important because when you build a tree, you have an outgroup, which puts all the other sequences into context. So I think that's enough for just a little tree here. We could just select all, and then we have 100 sequences in our tree. That would take a couple more minutes to generate. So here we got 12 sequences selected. So I will download that. And FASTA is the correct format to download this in. And we will open it with Notepad. And I got to go over here, and we have the Notepad shared. Okay, cool. So here's the FASTA. And you can see that when you get a FASTA from GenBank, it's got a really nice, a really long name in here. And a lot of the stuff isn't needed. You can like trim out the name of the gene because we know this is all the same gene anyway. And a really cool thing to do here is to put the location. So what I would have to do is look up this accession number and put the location. And when you have locations in your phylogenetic tree, that makes it much more useful. But it's also very time consuming to put in locations, so I will not do it right here. Um, but if I was going to make this tree to upload to the Facebook to share with my friends or publish in a scientific paper, I'd definitely put in locations because you get way more information out of it. Um, so you can see we have the bracket, the greater than sign, the name, the sequence, the greater than sign, the name, the sequence, and that just keeps repeating until the end of the file. What's not in this file yet is the sequence that I just generated because that I just submitted to GenBank and it won't be in there for a week. So I need to add in my sequence. So on a new line, just up at the top, we'll do our greater than sign, and this is Hygrostomi flavescens, and this is when I collected in Dalton, Michigan. And throw that back on the clipboard. Over here. And okay. So that looks good. And now we have to save this as a .fas. If we don't put in, if we just save it as a text file, that's going to make the other programs think that it's not a FASTA file. So this is hygrostomi.fas. And now we have our FASTA file. And now we have a lot of ways to generate phylogenetic trees. People have been doing this from sequences for about 20 years, but they've been generating phylogenetic trees using other character states that are not sequence-based for 50, hundreds of years, really. So in the last 20 years, they've developed a lot of algorithms for making phylogenetic trees. And what they all have in common is that they try to use math to calculate how all the species in your tree evolved. So the easiest way and the quickest way to make a phylogenetic tree is to use a website called phylogeny.fr. So if we go to phylogeny.fr, this is a website in France. And the really cool thing about this is it has a one-click mode. And with one-click mode, it does all of these different steps for you. And all you have to do is upload the FASTA, and then it'll do an alignment, so it aligns everything, and then it calculates the phylogenetic tree, and it visualizes it and does everything. So this was called hygrostomi.fas. That's the file that all of my sequences are in. That's that file that has 12 sequences. So we hit submit here into the one-click mode. And what it's going to do now is an alignment. So if you look at this graphic, you see how it's aligning everything vertically. It didn't take long because we didn't have very many sequences. So now it's on to calculating the tree. But when you align all the sequences vertically, then you can directly compare each position, each base, and actually do a meaningful comparison. So here's my tree here. First thing I'm going to do is make it a little bit bigger. And so here it is. And oh, it looks like we have the notepad on top. So I have to put this down so you can see what I'm doing better. Firefox needs to go up. Okay. So here it is. My sequence here is this one here, Hygrostomi flavescens, Michigan, Dalton. And everything that's on the same part, the same branch here, is the same species. This one here is probably a dirty sequence of the exact same thing. But this one here is my sequence from California. So you can see that this is the closest sister species, is this species from California that looks the same but isn't. And here, if I had put all of the locations, here you would be able to see where else in the world my exact same species occurs. And it's all over the East Coast, Smoky Mountains, all sorts of places. And then Chlorophyna occurs both in North America and in Europe. And you can see that, these other species here. One thing that we can see is that Hygrostobe mollis and Hygrostobe citrinovirens are the same species. They got the same sequence because they're exactly the same right here. And then this one, Chlorophanna, branched off from that. At least that's how the mathematical algorithm thinks that these things all evolved. So this is an algorithm called maximum likelihood. And then here, these numbers are bootstrap support. And bootstrap support is it's calculating how likely it is that it got the branch in the right place. So bootstrap support over zero point 0.7 is generally thought of as pretty good. So this one just barely makes it. If it's under 0.7, then the computer is really just guessing. And so it's just guessing where to put the branches. And so reality could be much different. So you definitely want higher numbers here. And one way to get higher numbers is to sequence more genes. And by sequencing more genes, you can have more confidence that these things are all going in the right spot in the tree. But this bootstrap support's not bad here, so this tree is probably correct. I will show you now. Let's see if we'll pull up. I'm going to show you what it looks like when you have a sequence that's bad. And that was, let's see, was it? Or not necessarily bad, but partially bad. Let's see. I think I have to add this over here to the stream. And we want this other one. Okay. So this other one is another trace I was looking at for someone yesterday. And this trace is a gene called the LSU, a large subunit. And LSU is cool because it's more conserved. So it's worse for identifying exact species, but it's better if you need to compare sequences from organisms that are not closely related. So ITS is very variable, and it's so variable that it's really good for species level resolution, but it's so variable that you cannot compare things in different genera and have decent bootstrap support. So, looking at this LSU trace, you can see it's kind of weird here. I don't really trust that this is correct. I think the first good peak that I see, is the C, but the one before it was an A. So I think what I stopped trusting is maybe there's a G in here or something. So I stopped trusting it here. So I'm gonna chop off that end, and now we're going to scroll through the sequence and see how it is. And you see this data looks really good. We have all these nice good peaks. They're all sharp. There's no weird background signal. Nothing. You never get two things in the same place. So just going through here, this looks really good. And then all of a sudden we start to see data that does not look good. And what happened here is we have a whole bunch of T's in a row. So we have 8 T's in a row. And what that does is it causes enzyme slippage. And enzyme slippage causes it off by 1 error in the sequence. So now we're starting to see we're getting problems because over here on the right, the peak here occurs again in the next one. So here we have a black G, and that's good. But then we see the black G again over here, and we shouldn't see that. That's the enzyme slippage. Again, here's an A, and then here we see the A again, and this is actually causing an error because we see this red is a T, but the A from this one overshadowed this T here, so this is a problem. We should change this to a T, and I always keep the changes lowercase so I know what exactly I changed. Here we see the same problem again, but this time the real signal won over because we know the fake signal is from the peak before it because it just slipped by 1. So I can go through really slowly and clean up. Here we have an A and the G in exactly the same spot, but I know that the A was a slippage from this, so this one's good. I can go through here and I can clean up this problem, but that's a lot of work to go through here and clean up, and maybe it's not even worth doing because very often you make mistakes here and you got to correct the mistakes later in BLAST, and it's really time consuming. So you can see this enzyme slippage problem just continues off until the end of the sequence. And here, Sanger can read about 1,000 bases, but towards the end, these peaks don't look so nice. They get a lot more rounded. You can't trust the data quite so much. And this data at the end here is clearly garbage. So I just highlighted all the garbage data, and I'm going to delete it. So, so that's a really important thing to do, is to keep the good data and delete all the garbage. So this sequence here is pretty good. So now we've seen how to build phylogenetic trees. I'm going to show you a phylogenetic tree that I built, and we will put Firefox back up on top. So here's a phylogenetic tree that I built of Verpa. And what I did is I got a whole bunch of sequences. Some of them I made at counterculture labs in Oakland. Some of them got emailed to me from people in other labs, and other ones I downloaded from GenBank. So the ones that I made, I put one color on, and the ones that people emailed me, like the different people, I put different colors, so you know like which color they came from, which person they came from, based on the color of dot there. And that helps me see which sequences are from GenBank and which ones I actually have the specimen available. So you can also see that I put the names on here. And so what I did is I wrote a program that looks up each record in GenBank and puts the name, puts the country, the location, into the FASTA file. And that program is available on GitHub in case anyone wants to use it. But you can see this phylogenetic tree looks pretty nice. And what we learned from this is that Verpa bohemica is pretty much one species, except for the one in Iowa and one in China and India. But so Verpa conica is pretty much correct. Like if you find a Verpa conica in North America, as long as it's not in Iowa, you probably are identifying it correctly. But Verpa conica, which is people used to think was one species, there's a lot of species here going under the name Verpa conica. Here's one, here's another one. So there are like 10 different species going under Verpa conica. So this is the kind of thing that you can see in a phylogenetic tree that you will not notice in BLAST results. So, just about finished. I want to show you a couple resources that we use for sequencing. So here is a Facebook group called Fungal Sequencing. We'll go to it. It's Fungal Sequencing. And Fungal Sequencing is a group that I created a few years ago, just to have a general place where people can discuss all different aspects of sequencing fungi. So you can see, like, here's pictures from my DNA workshops, and people are asking all sorts of questions about making their PCRs work, and just stuff like that. So, let's see, there's William Padilla Brown. He bought one of the Odin kits. He brought the big kit to do everything, and now he's sequencing stuff, and you can see he's getting pretty good results here. So just as a place to ask questions about fungal sequencing, that's a really good place to do it. And then I made another group, and it's called Fungal Sequencing Results. And this is just a place to publish results. So, publishing in scientific journals is definitely cool, but it's a lot of work. And I wanted a place where I could just put stuff up like really quick. Maybe it's not a good enough tree to publish in a journal, but it's good enough to at least share with people. So we have fungal sequencing results for that. And here's Terry Clements in Arizona. She sequences a whole lot of stuff, and has discovered all sorts of new species from Arizona. And then there is a wiki, and unfortunately the wiki is down right now, but that's the first time I've ever seen it down. But if you look, here's the URL. It's wiki/DNA_sequencing. So if you copy down that URL, it will work tomorrow. And what I did in here is I wrote how exactly I do my DNA extractions, PCRs, sequencing, the electrophoresis, building phylogenetic trees, pretty much just the whole process. I just wrote down exactly how I did it, so if you wanted to start sequencing at home, you could just load up this Wiki page and just follow the instructions there. And then at the end, it has a list of all the equipment you need for sequencing, with places you can order it and the approximate price. So setting up a DNA lab in your house is going to be somewhere between $500 or $1,000. There's a couple of programs that people use to do this sequence analysis. And probably the most popular program is called MegaX. So MegaX is from megasoftware.net. It's Mega version 10. 10.1 is the newest, so you can download this for free. It's got Windows, Macintosh, Linux. And so this is a really cool program, and it does all sorts of things, but usually what I use it for is doing an alignment. So I'll use it to do I'll do an automatic alignment, and then I can verify that automatic alignment manually to make the alignment even more accurate. And then you can build phylogenetic trees, and it has a whole bunch of algorithms. It's got neighbor joining, which is super fast and sort of accurate, or it's got maximum likelihood, which takes longer to calculate, but is even more accurate. And you can do distance trees and all sorts of other sequence analysis stuff with MegaX. And then if you're a professional, you use a program called Genius. And Genius is really cool. You can view the chromatograms like we did with SnapGene Viewer, except you can view the forward and reverse read at the same time and compare them in real time, which is pretty nice. But Genius costs $500. But they got a free trial for 14 days, and even after that free trial is up, it's still pretty good for a bunch of stuff. So those are the two programs that I recommend. And then if you want to make professional grade trees, like for publication in some sort of magazine or scientific paper, you want to use some of this professional tree building software. And a very popular one is RaxML. And RaxML is a beast of a program. It is not easy to use at all. It does not have a GUI. Though some people have written some for it, it doesn't really make it much easier. But and it's very slow. Like with the RaxML, you might start your calculation, and if you have a lot of sequences in there, you come back in the morning to get your results. But it does a very good job, and it's one of the most trustworthy ways to make trees. And then it calculates the bootstrap value, and what it means is that it generates a tree 1,000 different ways, and then it scores how good that tree is. And so out of those 1,000 different trees, it figures out which one was the best, and that's how it calculates the bootstrap value, so you know how much you can trust the different branches. So if you really want to make a good tree, you can use RaxML. And then there's another one. There's another analysis method called Bayesian inference, and that is usually this program called Mr. Bayes. And Mr. Bayes, like RaxML, does not make any attempt to be friendly. It's not unfriendly, but it's very much command line driven, and it's definitely a beast, and it takes a very long time for it to finish. But if you're going to publish something, you might as well use some of these programs. Though really, I've used these programs a lot, and the tree that I get, my quick tree from phylogeny.fr, the one-click method, is almost always the same. So just for a quick tree for yourself, you can use that. And I think that's just about all I got for you. We're almost out of time. But if you have any questions on any of this, you can contact me. I'm Alan Rockefeller at gmail.com, or Alan Rockefeller on Facebook, or Instagram, or all those other things. Thank you very much for watching, and don't hesitate to contact me if you got any questions or comments about this.